Going to talk about incorporating recirculation effects into metrics of feeding performance for zooplankton. Zooplankton are a subgroup of planktonic organisms that cannot generate their own food and have to look for it in their environment. And the study of their feeding performance is important because it can explain aspects of their evolution and behavior. As an example, consider S. rosetta, which is a single-cell eukaryote found in marine and fresh waters that preys on bacteria utilizing fluid currents generated by its flagellum, as shown here. And it uses the fluid currents to bring bacterial prey to a filtering surface, as shown here, where the prey would be captured on the surface. S. rosetta goes through different life stages, including when it's attached to a surface, or when it's a single cell swimming in different forms. And interestingly, they've also been observed to be able to form colonies in the shape of rosette-like structures or chain-like structures. And this is interesting because if proven that through cooperation these cells can provide survival advantages, it could give us insights into origins of multicellularity. So several studies have examined the feeding performance in single cells versus colonies, trying to look for possible advantages. A common approach in these studies is to think of the net inward flow as a proxy for nutrient uptake. The idea is that nutrients are dispersed in the fluid homogeneously as passive particles, and through periodic beating of a flagellum or cilia, the organism utilizes flows to bring particles to a filtering surface. Whereas the fluid passes through the surface, the particles are captured. For example, in the case of S. rosetta, as the organism swims downward, it has these flows that bring the particles to this filtering surface shown here. So if we can compute the net inward flow using this equation, we have a measure for the nutrient uptake for the organism. And the important part with the equation is that the area integral only includes parts of the surface where the flow is moving inward, actually. So we have several studies using this approach for S. rosetta. For example, in Roper et al. 2013, they modeled the beating of the flagellum in the far field as the effect of a stresslet. And they use this spherical surface far from the cell and compute parts of the flow that goes inside this surface, shown in blue in this schematic. And they conclude that the feeding performance in colonies is better than feeding performance in single cells. However, looking at this schematic, you can see that there's no guarantee that all the flows going inside through these blue regions actually end up on the filtering surface. They can go inside and exit through these red regions without ever touching the filtering surface. So in a more recent study by Kierkegaard et al., they model each cell as a sphere and a thin straight rod representing the average geometry of the flagellum through one period. And they examine the flows on the actual filtering surface. Computing part of the flow that goes inside, the blue region in the schematic here, they come to the opposite conclusion that single cells have better performance compared to colonies. In a more recent work by Nguyen et al., they have the hydrodynamic effects of the filtering surface and they have the actual geometry of the flagellum throughout the period. And they compute the instantaneous velocities on the surface at all times, using that to get the average velocities and find the flow, part of the flow that goes inside on the filtering surface. However, since they have the instantaneous velocities, it raises the question of how to interpret this integral. You can either use the approach taken in the Nguyen et al. Paper, where they find the average velocities at all points on the surface, and obtain the parts that the flow goes inside, the blue region, and then integrate the flows on that region to get your measure of nutrient uptake. Alternatively, by changing the order of the integration, at any point in the period you can obtain this blue region and find the instantaneous velocities to obtain the flows on the blue region, and then integrate those values throughout the period. And it's not clear which approach is the correct one. We saw that to use the net inward flow metric, we need to determine what surface to use to be guaranteed that the flows on that surface are containing new nutrient particles, and we need to determine should we use time average or instantaneous velocities. But remember that the inward flow was attempting to measure new nutrient particles being captured on this surface. So the correct feeding metric should directly track new particles using a Lagrangian approach, as we do in this work. The method we use is the method of regularized Stokeslets. We discretize the surface of our cell with Stokeslets and find the forces on each one of them, which consequently allows us to find the velocity at any point in the space as a superimposition of the effect of those Stokeslets. And the geometry parameters that we use, we take them from the Guen et al. Paper for S Rosetta. As part of their results, they have the inward flow as a function of the capture zone length for S Rosetta. By capture zone length is the length of the filtering surface here. We are showing the original data in blue in these two plots, and the red is our attempt at regenerating that data using the average velocity metric, and the yellow is our results when we use the instantaneous velocity. And you can see that the average and instantaneous flows can be very different. We identify the reason for this difference to be the existence of recirculatory flows, and we also explain that because of the existence of recirculations, it is not guaranteed in general that neither of these two measures give us the correct value of nutrient uptake. Imagine the schematic here where we have divided the filtering surface into 4 area elements. On the first two down here, we have the flows containing new nutrient particles coming from somewhere far away, and the particles are captured on the surface, and the flow goes back to somewhere far away. On these two elements, average and instantaneous velocities are the same, and both metrics measure the correct amount of nutrient capture. However, on this third element, imagine that we have a recirculation within one period. Average velocity is 0 on this element, while instantaneous velocity changes through the cycle. And only average velocity metric measures the correct amount of nutrient capture, which we expect to be 0, because it is a recirculatory flow that does not contain any new nutrient particles. Finally, on this last element, imagine that we have a recirculation longer than one period. Average and instantaneous velocities on this element are the same, and neither metrics measure the correct amount of nutrient capture, which again we expect to be 0 because it's a recirculatory flow. So since the recirculations are important in our analysis, in this work we look at another organism that is known for generating recirculations around the cell, namely Vorticella, which is a bell-shaped organism attached to surfaces via a stalk, and it uses its cilia, as shown in the videos, to bring flows containing prey to the mouth. For the geometry of the beating cilia through the period, we use the data from this paper, and from our videos we determine the direction of the power stroke to be pointing outwards. We simulate the flow field for the Vorticella in the presence of a wall here using the method of images for regularized Stokeslets, and we confirm that it is qualitatively similar to our videos in the lab and what the reports in the literature. Also, I wanted to point out that while in reality we have the cilia for Vorticella beat with a phase difference, generating a metachronal wave, here in our work we keep the symmetry and assume that all cilia are beating synchronously. Our goal is to find the flows that end up on the Vorticella containing new nutrient particles. To do this, we obtain tracks of particles that are filtered by the cilia. We start with particles on this desired surface shown in green, and we assume that any particles reaching this surface from either direction are captured. We obtain the trajectories of particles that end up on this surface by starting from their final position and propagating those trajectories backwards in time. And as we do this, for each trajectory, we continually check to see if the trajectory would pass the desired surface sometime back in the time or not. Because if it does pass the surface in the past, it means that on that part of the surface, the flow at the current time does not contain any new nutrient particles. Also remember that our flow is not steady, but rather periodic. And the path lines of particles at one time through the period and at one point on the surface are not the same as path lines of another particle that ends up on the same point on the surface but at another time in the period. So we need to repeat this process for all times through the period. And when we are done, we know for any given time, we exactly know on what parts of the surface we have flows that contain new nutrient particles. And this allows us to introduce our own metric for feeding performance. In this in this figure, the blue curve shows um, uh, the metric where we use the instantaneous velocities to capture the flows, as we discussed in the S Rosetta, and the red is our metric, which is only includes the flows that contain new nutrient particles. The dashed lines show the average values for these two through the period, and the green line shows the metric where we use the average velocity. And as you can see, our metric is much smaller than the other two, and it indicates that most of the time the inward flow does not contain any new nutrient particles. Finally, we might want to find out a scanning region where all the flow going inside that region would end up on the surface and contains new particles. And the reasons for doing this is similar to the Roper paper, we might have a model for the far field flows, and we want to know on what region in the far field we have flows that end up on the surface containing nutrient particles. Also, another reason could be that in experiments, it's easier to resolve flows farther from the cell compared to flows very close to the cell, as in this experimental work by Nagai. So since we have our trajectories, we can make this a 3D structure, and the plot here shows the flows that we have computed at different cross sections of this structure as a function of the x position. And these values are similar to our metric shown here in red for Vorticella. So to conclude, we showed that the inward flow metrics are not a good way of measuring nutrient uptake, and instead it should be determined which flows actually bring new nutrient particles to the organism. And this could also be affected by having active swimming prey particles instead of passive particles, and also by the diffusion of the particles, which we did not discuss in this work. Thank you.